everyone, and welcome. So today we continue our discussion on the link between genetics and neural anatomy and function. Last week, if you remember, we learned that memories that affect behavior can cross several generations through epigenetic transfer in flatworms. And today we'll hear about the genetic code itself and how information affects neural diversity. We're happy to have Professor Oliver Hobert with us. Oliver will tell us about Homeobox genes, which are the genes that regulate development and morphogenesis. And will tell us about how they encode neural diversity. Oliver is a professor of biology at Columbia University. His lab is part of the Neurotechnology Center in the Zuckerman Institute at Columbia. His lab studies the genetic and evolutionary origin of neural diversity and morphology. Thank you for joining us.
Thank you very much for the invitation, and thanks for the introduction. And it's a real pleasure also follow up with Les, who was a postdoc in my lab, and to see him become such a prominent person in so many regards. So anyway, I will continue discussions. You heard about C.elegans, of course, and I want to tell you more about the C.elegans nervous system today. And I want to tell you about how we approach the problem of how an organism generates the extent and the amount of cellular diversity that characterizes any brain, including that of C.elegans. So C.elegans has 302 neurons that fall into 118 different classes. And what my lab is trying to understand is how these 118 different classes are generated. And I say 118 different classes, which are all anatomically defined as unique classes, because we're not just interested in some part of the nervous system. We really want to look at the entire nervous system with the ultimate hope of extracting some common organizational principles for how not just some part of the nervous system, but the entire nervous system is becoming genetically specified, and how cells in the brain of C.elegans assemble into circuitry. Okay, so if you think about what it means to have diverse cell types in any nervous system, you can think about this from a very biochemical, biophysical way, in the sense that you can visualize or think about different fully mature, fully integrated neurons in the nervous system as expressing distinct set of molecular machines that assign specific functional and biophysical properties to a neuron. Neurons are different from one another because they express different sets of, let's say, ion channels, different neurotransmitters, different neuropeptides, different neuropeptide receptors. So essentially, if you want to really conceptualize neuronal cell type diversity, we're essentially talking about a gene regulatory problem. We're talking about the problem how distinct cell types turn on different sets of genes. And I'm simplifying that here in this manner, which really lays down the very simple questions that we need to understand. We need to understand what exactly are all the genes that are expressed in an individual neuron type to endow it with its specific functional structural features. And from the perspective of genes, we need to understand how the expression of individual genes in the nervous system is regulated. As I'm sure you all appreciate, there's no such thing as genes that are expressed just in one neuronal cell type and nowhere else. That doesn't exist. What you rather have are genes that are expressed in multiple cell types, but in a highly cell type specific manner. And it's this combination of on-off profiles that essentially is sort of the fundamental unit of cell type diversity in the nervous system. So this is basically what we need to decode here in order to understand how distinct cell types are being generated. Okay, so now if you take a step back here and think about an individual neuron class and think about the very different molecular processes that go on in a neuron and the different proteins, the different machines that execute these different functions, you can think about, if you think about their regulation, how do they become turned on in specific cell types? You can think about it in two fundamentally different ways. You can think about different biochemical entities of a neuron being regulated totally independently of one another. There could be different gene regulatory factors, different transcription factors that some of them dedicated to turning on the receiving end here of whatever information a neuron receives, other transcription factors being required to select a specific neurotransmitter, and so forth and so on. So this is sort of one extreme way to think about that. The other extreme way to think about this is a seemingly much simpler way, in which maybe all of the very different biochemical properties of a neuron is actually regulated by the same transcription factor, right? Which would be pretty amazing if you think about this. You know, it would mean that there's some sort of master regulatory mechanism here in place that coordinates these biochemically quite distinct features of a neuron. So how can we distinguish between these models? Well, these models have been very clearly been distinguished in C.elegans over the last 2 decades or so. And the answer to this problem here, to these 2 different models, is very clear. And let me tell you where, and let me just give you a very brief historical perspective of that. The person who really cracked open the problem conceptually, not knowing what would come out of it, of course, was of course Sidney Brenner, who did the first mutant screens in C.elegans. And what he identified were mutant animals that displayed specific behavioral defects. This is what he was looking for. Subsequent work then showed what kind of neurons are involved for executing these specific behaviors, either because they are the critical sensory neurons or critical downstream interneurons that process the sensory signals. And what was noted for a whole bunch of these cases here, that the mutant phenotypes observed in these mutant animals was identical to the microsurgical removal of the entire cell. Like, for example, if you microsurgically remove this one single interneuron class, doing what I'm basically doing now, just with much higher laser power, you laser ablate this neuron class, you'll get very specific behavioral phenotypes, and TKX3 mutant animals look identical phenotypically. These genes were then cloned over the years by several labs, including my own, and what was then found and appreciated is that these genes all code for transcription factors that are expressed in these specific neuron classes. Let's go into 2 examples here. This is an example that Marty Chelfie at Columbia studied in quite some detail, and this is an example that we studied. These turned out to be transcription factors that indeed do not regulate just one specific functional aspect of the neuron, but they co-regulate every single molecular feature of that neuron that we were able to get our hands on, so to speak. Right? So any gene whose expression we could measure at that time and is expressed in that mature neuron class fails to be expressed in animals that lack this transcription factor. And the same for this different neuron class here. What you also already notice in this scheme here, very important principle that is not unique to C.elegans. I would never claim we discovered that. It's sort of a conceptual expectation that has existed in many cells already before, that transcription factors usually never work in isolation. An individual transcription factor has too small a DNA binding domain to encode enough specificity to selectively identify its target in complex genome sequence. So transcription factors usually always work in combination. And these are beautiful examples of that, because in those cases, it was found that those 2 transcription factors each are essential for expression of these target gene battery, and they're each essential because they work by the principle of cooperative DNA binding. Look at these 2 transcription factors. They're actually each expressed in a bunch of different neuron classes, but they uniquely overlap in their expression in that neuron class. Hence, in this neuron class, and only in this neuron class, they can heterodimerize, and only as heterodimers are they able to bind this little sequence motif that is present in all of those genes, for example here, neurotransmitter synthesis pathway, right, that are expressed in that specific neuron class. Okay? So this combinatorial theme of transcription factor action, I want you to really remember that. It's going to be a recurrent theme throughout my entire talk. So in many cases, people would have found something like this, been very happy with what they found, and moved on to study something else. But what we really wanted to see over the years is this master regulatory principle. The idea of having these transcription factors, which we call terminal selector transcription factors, because they select the terminal identity of a neuron class. We wanted to see, does that coordinated regulatory principle apply for the entire nervous system of C. elegans? The way we approach that problem is to make use of a resource that the entire C. elegans community has built over the decades. What they have done is various different labs throughout the world, their favorite gene of interest. Whenever in C. elegans you study a certain gene, you will one day, sooner or later, fuse your gene that you work with to GFP, make transgenic worm, look where your gene is expressed. This data will go into a database, and you can extract this information from a database, and you will see that for almost 1,000 genes, GFP reports have been described where people have really bothered to look where in the nervous system are these genes expressed. So each blue dot indicates an expression pattern, and each line here, each row is a neuron, 302 neurons, 118 different types. And you will see for each neuron the expression profile here of these reporter genes. So this is sort of a pre-signal sequencing molecular fingerprint of the entire nervous system. Yes?
So up till now, everything you've described has been binarized. And I'm wondering if the data, the GFP, I can see how that's binarized. But when you have the opportunity to look higher resolution, whether the data is also binarized at the single cell level.
It's a great question, and it's not. There you can clearly see it's not. What I mean by it's not, if you really quantify with modern day technology, that includes things such as GFP insertion into the genome of a specific locus and then really quantifying gene expression in different cell types, you will usually always see different expression levels. That's not to say that these different expression levels really have some informational value attached to them. Right? It could really be an artifact or a secondary consequence of whatever the transcription factor combinations. Right. And this is really hard to prove, that levels really are important. There's one example where it has been explicitly shown, actually on the level of the transcription factor, where the expression level of a transcription factor in two different cell types that are different from one another determines actually alternative fates. So yes, with a pure binarization, I'm definitely oversimplifying and ignoring additional levels of complexity.
But you do believe that this represents reality more than the nuance?
It's a really good first pass. Right? You can surely really refine that, and think about a more fine-grained neuronal classification based on levels. But I would call this a first pass quantification in binarization. And thank you for disrupting me. So any questions, please ask as I move along. I really appreciate that. Okay, so what we then did over the years is to make use of these GFP reporters, and we did either genetic screens for mutants, and I won't bother you with the details. I also won't bother you with any sort of technical stuff here. I'll just give you the punchline. The punchline is that we identified many different transcription factors that act in many different neuron classes here to specify their identity in this coordinated regulatory manner. And the way I'm summarizing this data here, the way you have to read this table, again, a binarized version here, where you basically see here these two blue dots. That means these two transcription factors here, they are jointly required for this blue neuron class, a dopaminergic neuron class, to be functionally specified. Okay? So meaning, yes, we do believe that this idea of combinatorial codes of transcription factor that specify unique cellular identities is operating throughout the entire nervous system. Now, I want to just to drive home this point, I want to address, right now we've taken sort of a very cell-centric view where we've asked here about individual cells, what transcription factors do they use. I want to actually go back to this original scheme here and look at now a single gene, not from the perspective of a cell, but I'm asking now, well, based on what I told you, that each neuron has a distinct combination of transcription factors that turn on a gene in some cells here, and it's not turned on in other cells. What does it mean for an individual gene? Let's go through an example here. Let's go through the key gene that you need to express to become a glutamatergic neuron. It's actually sufficient to have one gene to become glutamatergic. This is a gene that encodes a vesicular glutamate transporter. If you express that, you'll be able to take up glutamate that every cell has, and this is a vesicular transporter. It will be brought to the synapse, and you'll spit out glutamate. So every neuron that expresses in C.elegans or in the vertebrate nervous system, any one of these V-glut genes here is a glutamatergic neuron by definition. So, and as indicated here in yellow, there's many different classes in C.elegans, neuron classes that use glutamate as a neurotransmitter. There are very different neuronal classes, sensory neurons, interneurons, even some motor neurons are glutamatergic. And based on what I told you so far, you would predict that this gene, to be turned on in different neuron classes, it would need to be responsive to different combinations of transcription factors in different neuronal cell types. And if this were really true, you can experimentally test that. And in C.elegans, you can very easily test that by taking that gene, this locus, this non-coding region, you fuse it to GFP, and then you mutate these predicted binding sites for these different combinations of transcription factors, and you should now see loss of V-glut expression only in very select neuron classes. And this is exactly what we see. So we take the entire V-glut locus here, fuse it to YFP in this case. These are all the 38 glutamatergic neuron classes, and this is now a totally random chopping of the promoter in different fragments, and you will see that different cis-regulatory elements drive expression in different cell types. You can do a more fine-grained mutational analysis. You will then mutating this one very small predicted transcription factor binding site, you lose expression exclusively in this one neuron type. And that's again totally consistent with this idea that you have different combinations of transcription factors in different cell types that turn on or not, if the combination is not there, that specific neuron, that specific identity feature of a neuron in different neuron classes. And again, yes, please.
Is there no like epigenetic layer for this regulation? Like that, are these transcription factor binding sites accessible?
It's a very loaded question.
Sorry.
You know, the reason I call it a loaded question is that to me epigenetics is always sort of insinuating that there is a code there, a specificity code that's independent of transcription factors, and that's clearly not true. I mean, if you think about any type of chromatin modification, any type of accessibility issue, there has to be some specificity code, some other transcription factor that generated this accessibility, for example, or that it must exist here. I'm sure there are going to be some earlier acting factors that generate accessibility of specific regions in the genome. They have not come out of our genetic screens, presumably because those chromatin factors are, having very pleiotropic functions. So I don't deny the existence of it, but these are sort of the real specificity components that are really, in a DNA sequence-dependent manner, are absolutely essential to turn something on. There's clearly permissive environments that are also contributing to that. Okay, and just to basically show again what I sort of showed you before already, again to fulfill this prediction here, indeed you get very selective effects if you mutate specific transcription factors. Here's one transcription factor. Let's look at this guy down here. This is a V-glut expression in a bunch of head neurons here, and you eliminate one of these transcription factors here, you will lose exclusive expression of that thing in a specific neuron class, in this neuron, but not in the other neuron classes. Right? So if this highly modular composition here is regulatory information, modular regulatory information that is being interpreted by these cell type specific combinations of transcription factors. So I want to now move back to this table, and I want to sort of stare at this table a little harder, and I'll do that twice with you today. Staring at the table like this and sort of trying to extract some principles out of that may not hopefully tell us something about the C.elegans nervous system, but really may tell us something about how nervous systems evolve. Now I'll come to this evolution aspect at the very end of my talk, but I want to first here bring home a point that you could have only seen if you had very good eyes, which is that the majority of transcription factors that we found to be involved in neuronal identity specification are actually members of a specific family of transcription factors encoded by Homeobox genes. Now I want to give you one disclaimer right away. There are also non-homodomain transcription factors that are involved in neuronal identity specification. When they are, they usually work together with homodomain transcription factors. Here's a guy, this green box. This guy is required to specify that neuron class, but look, it works together with a homodomain transcription factor. So they really seem to predominate the landscape of neuronal identity control, that specific transcription factor family. So I want to give you a little bit of background on those guys. You probably heard about Homeobox genes from, if you're not really a molecular biologist, you may have heard it from your biology undergraduate classes. This is sort of the textbook example of a success story in developmental genetics. These genes were initially identified actually at Columbia. This is the famous fly room here at Columbia by Kelvin Bridges, who identified a mutant by thorax that was then characterized by other people, including Lewis and many others. And the punchline here is that there's a specific subclass of Homeobox genes, the so-called Hox cluster genes, that are organized in a funny way in the genome. They're all clustered together, that are providing positional information in a developing organism along the anterior-posterior axis. So this is what probably many of you have heard about in developmental biology classes. But I want to point out that this is a very specialized and in fact derived class of genes. Derived meaning this is not their ancestral function to do something along the anterior-posterior axis. They're actually just a small part of a much larger family of these transcription factors. Animal genomes have anything on the order of 60 to 200 or so Homeobox genes encoded in their genome. And what I want to address with you is the question of whether those Homeobox genes can also be assigned to a very general function, like the Hox cluster genes in segmented body structure, but maybe we can assign an ancestral function of those genes to neuronal identity control. So Molly Riley, a graduate student in the lab, set out to do that, and given this lots of preliminary functional data we had about the Homeobox gene, or the importance of Homeobox genes in neuronal identity control, she asked, can we make Homeobox genes be responsible for every single one of the neuron classes in C.elegans? And what she did, to address this question, she took every one of the 102 Homeobox genes, tagged them with GFP, and then looked where are these genes expressed. And I want to sidetrack a little bit into the technology here, because this is going to show something that's actually aesthetically quite pleasing and beautiful. Let me set up the problem first. If you do that, and if you look at gene expression in C.elegans, you see these beautiful green dots, and even though the nervous system is very simple, you cannot say, based on position alone, oh, this has to be neuron ABC, this is neuron DEF. You can't easily do that. It's very hard to really be sure which cell this is, which cell this is. So what Ed Yemeni, a postdoc in the lab, did, and I'm happy to take you through the details afterwards or now if you insist, he built a transgenic worm in which each individual neuron class is color barcoded in a totally stereotyped manner. Color barcoded in a way that every neuron has a different combination of fluorophores relative to its neighbors. I'm not saying there are 302 different colors here. I'm saying there's enough color here that I can distinguish every cell from its neighbor. Now the cool thing about this fluorophore mix used here, he used the whole palette of fluorophores except GFP. So what we can do is, we can take these GFP reporters, cross them with that strain, and based on where the GFP signal is, we can say, based on the color code, oh, this has to be this neuron. And only because we had this tool available, we could do this type of gene expression analysis on a scale that spans the entire nervous system. I also want to point out, and this is particularly meant to you guys who do single cell sequencing. What you do, guys, is great stuff. It's wonderful, right? But never forget that you only look at transcripts. That's not what we're doing here. We are looking at proteins. And as many of you know, there's lots of stuff that has to happen before an mRNA is being made into a protein. So this is data that you would not yeah, then in fact we did single-cell sequencing of the entire C.elegans nervous system. It was published last year, and there's really interesting discordances between the single-cell expression profile and these protein expression profiles, right? So this is not just, okay, yes,
What
please.
you measure here is the expression of GFP, not of the host gene.
Sorry, say it again?
What you measure here is basically the expression of the GFP, not
The GFP fused
fused to the
homodomain protein, yes.
Okay.
Yes. It's important. It's not split in any way. There's no sort of
So it's
fancy.
one ORF, basically.
Sorry?
One open reading frame.
Exactly right.
Yeah.
Exactly right. Okay, so what's the punchline of this analysis? So first of all, what was quite striking is that this protein family shows actually throughout the nervous system a pretty sparse expression. So the majority of these transcription factors are expressed in the nervous system, except some of them, and I'll talk about them actually for quite a bit here, these ubiquitously expressed ones. But let's look at those guys first here. Quite sparsely expressed, usually on the order of every neuron class expresses about anything between 1 and 10, but usually around 3 to 5 Homeobox genes in combination. But most amazingly is this, and this is a slide where you just have to trust me what I'm saying. Every single neuron class in C.elegans has a unique combination of homodomain proteins expressed. So you will not see any line here that is the same. So meaning, you can basically reduce complexity of neuronal cell types in C.elegans. And again, those were initially defined purely based on anatomy and synaptic connectivity. You can reduce that to a single descriptor. And that descriptor is the combinations of homodomain proteins that are expressed in an individual neuron class. I do want to point out there are papers now emerging, and of course there's no paper yet that looked at the entire nervous system, at the entire central nervous system, for example, of a mouse, or let alone a human, and not even in flies. This has been achieved with a significant enough level of depth, but in those cases where people have looked in certain parts of the nervous system, here are two papers that I give you as an example, and both of these papers noted, when they looked at whatever their favorite part of the nervous system was, is that actually the Homeobox genes, and this is transcript data now, this is not protein data, but nevertheless, the Homeobox genes were the gene family that best discriminated between individual neuron classes. Okay, so I've told you sort of the story in reverse. Normally people show you expression profiles and then they want to prove the functional relevance by then doing some functional analysis. I've actually started with functional analysis and now get you basically expression patterns. So when you ask me about the functional relevance of these codes, I will of course just refer to what I told you earlier in this table, that yes, we have identified, we have characterized these many Homeobox gene mutants for having a function in defining the identity of these neuron classes. But again, we have the ethos of the C.elegans community is not to sort of stop half the way or even 80% the way. We really are going now through every single neuron class based on these predictions and looking whether we can indeed find for every single neuron class through mutant analysis a function for Homeobox genes. And this is where it currently stands. We're almost there. We have clearly not, 100%, yeah, right. And I hope in a few months from now we'll have this get closed here. Okay, so what I want to do now is I want to again stare at this table with you, and I'll take you through 3 examples of these functional analysis that taught us really interesting level, and I want to call them sort of meta levels of understanding of how the nervous system is being specified. And the first one is seemingly the most trivial one. You'll see here that there's a whole cluster, a whole bunch of Homeobox genes that turn out to be expressed in every cell of the nervous system. And we saw that, when we saw that, we were very excited about this for the following reason. And again, if you're not a molecular neurobiologist, you may not have thought about this, but if you are, it's really a stunning lack of knowledge that we had for decades, and this is the following. I'll frame this in the context of the mutants that we studied here. When we have these transcription factors, these homodomain transcription factors that specify the identity of individual neuron classes, what we always find consistently from cell to cell to cell. We take it away, we lose these neuron-specific identity features, but the neuron is still normally always generated, and not only is it generated, it usually always still expresses so-called paneuronal features. So paneuronal features are, of course, those proteins that are expressed in all cells of the nervous system and do really important stuff. Specifically, if you think, for example, here at synaptic vesicle cycle, right? That's very important. That's something that every neuron does. But stunningly, even though this is extremely well studied, Nobel Prizes have been given for how these synaptic vesicle proteins work, nobody knows, until now, how these proteins are regulated. How are these proteins, or how are these genes just expressed in the nervous system and nowhere else? And what our genetic analysis in C.elegans has shown, and again, a few studies in vertebrates had shown that as well, that if you remove the individuality of a neuron by taking away these identity-specifying transcription factors, you usually leave that stuff always completely unaffected. This is important because it tells you that a neuron, if a neuron acquires its functional features, you can genetically separate its individuality from just being a generic neuron. Right? So how are these features regulated? I'll tell you right away that this is a very painful history, painful to me, but particularly to those students and postdocs in my lab who've spent many years screening for mutants in which they took these synaptic vesicle proteins, fused them to GFP, and looked for mutants in which a GFP fails to be expressed. And the result of these screens was not only nothing, it was even worse. We got mutants out that we thought were it, but then they turned out to be some weird, artifactual, transgene regulating, really painful stuff. So it's something that we have been annoyed by, and that's why we became so excited seeing these paneuronally expressed Homeobox genes. And even more excitingly, they all fell into one family. A specific family of Homeobox genes that are characterized by the presence of another domain called the cut domain. It's not exactly clear what it does. It may be involved in DNA binding, may also be involved in protein-protein interaction. But the punchline being is that two of these family members, say 44 and 48, are nicely expressed in all cells of the nervous system and nowhere else. And I'll tell you also right away that 4 additional members are actually ubiquitously expressed. So meaning every neuron expresses 6 of those guys. And this made us really happy because if you look at these paneuronal genes, these are all genes here that encode for proteins in the synaptic vesicle, in some aspect of synaptic vesicle biology or other paneuronal features such as proteins involved in neuropeptide processing, which are also ubiquitously expressed. What you can see is based on chip data that we had available for these cut homodomain proteins, you find very nice binding sites for those guys. So that is great, wonderful. Yes?
Can I ask something? Yeah. You talked about the spatial difference in, or maybe in cell type specific expression pattern. But what about the expression in time? Is the Homobox gene expression change throughout the life of the worm?
Yeah, very important question. The feature of all of these identity-specifying transcription factors, that holds for the paneuronal ones and the neuron-type-specific ones, starts after cells exit the cell cycle and they're maintained throughout the life of the animal. And not only are they maintained throughout the life of the animal, this being C.elegans, you can do fun genetic gymnastics and remove the function of that protein long after the neuron has differentiated. You can do it even in the adult. And guess what you see? If you do that, the neuron will lose its identity features. Right? So those genes, very important point, are not just required to initiate a terminal differentiation program, they're continuously required through autoregulation, that's how they maintain themselves, to maintain the differentiated state of the neuron. Okay, so having this binding site looked great. We can take these genomic fragments that have the binding site for these cut homodomain proteins, fuse them to GFP. We see paneuronal expression, looks great. We can do it even more sophisticated. We can tag these paneuronally expressed genes with GFP using CRISPR. We can, again, use CRISPR to mutate those binding sites, and what you'll see, lo and behold, is a reduction of expression of gene expression. Now, I know you guys, many of you are quantitatively thinking, and yes, I'll be now very quantitatively arguing as well. Now, satisfy you more as in the next couple of slides, you do not see a loss of expression. You see a reduction of expression. I'll come back to that, right? But anyway, it looked really promising in terms of it has to be these cut Homeobox genes. And of course, what happened is if you did the mutant analysis, initially it was painful again, but only for a short amount of time, because now with CRISPR you can easily build sextuple mutants, right? And that's what we needed to do in order to see the strongest possible phenotypes on pan-neuronal gene expression. If you just mutate individual genes, not much is happening. That's why we never got from other genetic screens, right? But the more you remove, the more you reduce pan-neuronal gene expression. Again, this is not just shown for one gene. The quantitative details look different for different genes. In this case, there are actually some genes where individual or double mutants give somewhat of a phenotype, but the overall theme of the sextuple giving the strongest phenotype is always the same, no matter what pan-neuronal gene you look at. As you can imagine, if you lose your pan-neuronal stuff, you're not a happy camper, and they are not happy campers. You can measure this in a variety of different ways. Here's a response profile to a certain drug that measures the secretion and the paralyzing effect of acetylcholine. Again, I won't bother you with the detail. Punchline is individual mutants, double mutants, not too much of an effect. Only if we remove all of those guys, we get a pretty much a complete unresponsiveness to the specific paralyzing drug. So this is a really important experiment now. This is again looking now at a different behavioral readout. It's the ability of the animal to move, and as you can imagine, they don't move very much if you cannot do regulated secretion of neurotransmitters. But what I'm showing you now is you can actually now go back into these sextuple mutant animal, and you can put back in any one of the 6 genes. It doesn't matter which. And in fact, you can even take a human homolog of that gene, put that back in, and you'll mutate you'll rescue the mutant phenotype. So what that says is it's really gene dosage that's important. It's not what type of family member you are. Pretty much anyone can do the job, but you apparently need to have simply enough protein in the cell, right, to activate pan-neuronal gene expression. And I will now wrap in the terminal selector stuff, these identity-defining transcription factors, and come back now to this quantity, to this levels issue here. So I've told you before, and I stand to that, it's really true, if you take away individual neuronal identity-specifying transcription factors that specify a unique neuron's identity, I told you that has no effect on pan-neuronal gene expression. And that's actually quantified here. Okay? But now if you take a sextuple mutant of these cut Homeobox genes, where you still have some pan-neuronal gene expression left, and now you remove, in a septuple mutant, that terminal selector transcription factor, you further reduce, if not completely eliminate expression of pan-neuronal gene expression. So this is the take-home message, the punchline here. What you will see here is that you have neuron type specific genes being regulated by these combinations of identity specifying terminal selectors, homodomain transcription factors. They also contribute to pan-neuronal gene expression, but what you really need for pan-neuronal gene expression is enough dosage of that class of specific homodomain transcription factors. And this is the point where I want to start speculating about evolution. The way you can think about this is that pan-neuronal gene expression is something that is extremely robustly regulated. Now think about what neuronal diversity and the evolution of neuronal diversity, if you think about evolving more and more complex brains with greater and greater cellular diversity, what that means is, it always means that you play around on a theme, right? Evolution basically selects for new combinations of genes that are expressed that give you a new cell type, many cases not going to be useful, not going to be having any adaptive value, right? And you achieve this by this relatively simple regulatory logic that you can envision is very amenable to the gain or loss of binding sites for these terminal selectors, right? So this is something that's actually somewhat labile, right? But here, you can't screw around much with this. You know, once you're locked into being a neuron on sort of an evolutionary time scale, it's very difficult to not be a neuron anymore. So this is one way to think about maybe how neuronal cell type diversity further and further increased through this type of regulatory architecture. Okay, so in the last part of my talk, I want to now discuss another intriguing theme. I hope this gets particularly sort of the circuit systems crowd more excited here. And this is again involving staring at those tables here. And again, it looks just like a colorful, fancy map here. But I want to ask now a very simple question to you. At first, a simple question, which is the following. So combinatorial codes of these transcription factors, right, different combinations, different cells, of course implies that one and the same transcription factor is used again and again in unique combinations. But let's think about an individual transcription factor. And I'm highlighting one guy here. Is there sort of any commonality, anything shared by those neurons that use the same transcription factor for their neuronal identity specification? Again, this transcription factor works in different combinations. But if you look at it from the perspective of this single transcription factor, it's expressed here in 20 neurons. What about those 20 neurons? This is a particularly simple and obvious case. It turns out that those 20 neurons are actually very special neurons. They're all heavily interconnected. And in fact, they form the enteric nervous system of the worm. The enteric nervous system in C.elegans, as well as in any higher vertebrate, is a self-contained network of cells that acts autonomously to generate this peristaltic movement of some epithelial tissue, the gut, to move food through your digestive signal here. And this entire circuitry, right, co-expresses as one homodomain transcription factor. And that homodomain transcription factor, I won't bother you with the details, suffice it to say, you look at lots of molecular markers that are expressed in these different neuron classes, they all fail to be expressed in the mutant. So that transcription factor is required to specify their identity. And again, it's required to do so in combination with other homodomain transcription factors. This is where our current mutant analysis stands, so no surprises here. But what I want to get to you now is the following question. It's a simple question that I like to see paraphrased by somebody else. So this is something that people have been thinking about for quite a while, studying gene regulation in the nervous system. People have actually appreciated for a long time here that there appear to be a common strategy to generate neuronal diversity across the central nervous system, where combinatorial expression of different transcription factors specifies blah, blah, blah. So everything I told you so far. But here's the interesting question. However, an open question is whether this process also encodes a molecular identity responsible for connectivity, for assembling neurons into circuitry. Meaning, is that transcription factor, C34, that is expressed in all of these enteric neurons also required to assemble these neurons into functional circuitry? Is it just regulating some genes that give it some biophysical properties, or is it having this non-autonomous function of assembling cells into circuitry? And the answer to that is, it seems to indeed have such a role, because if you look here, this is actually quite a sophisticated, both simplistic and sophisticated way to look at circuitry. What we're looking here is actually all of the synapses here in this enteric nervous system. These synapses are all made in a neuropil called the nerve ring of the pharyngeal nervous system. This is where all these synapses are formed. The processes of these neurons, they run all along here, right? But if we look down at these mutant animals, synaptic circuitry is totally messed up. Okay? Let's take this a step further. And let's now move outside the enteric nervous system, and let's look at this hairball here. Any connectome, of course, is a hairball. And the question is, can Homeobox genes somewhat break this hairball, this whole animal connectome, into smaller units? And do these Homeobox genes have a role in instructing the assembly of these smaller units into circuits? This is work by Emily Berkov, a graduate student in the lab, and I'll tell you about another transcription factor, of course, a homodomain transcription factor, expressed in a bunch of neurons here in the main head ganglia of the worm, the brain of the worm. You see here all the expressing neurons. There's really no relationship among them. They are cholinergic, glutamatergic, peptidergic neurons. They have no lineage, no shared lineage history. They come from very different branches of the lineage here. But look, all of those UNC42 expressing neurons assembled into circuitry. They're more heavily interconnected to one another than they are to the rest of the nervous system. UNC42 acts to specify the individual features of neurotransmitter identity, all of these molecular features that I told you about. But we also, again in this case, think that UNC42 is required to assemble those neurons into circuitry. We say that based on data that I'm too ashamed to show you. I'm too ashamed to show you that data because we did something that sounds very heroic. Every person who does EM connectomic works thinks they are a hero, and they are, because they spend many years on reconstructing the connectome of usually a single specimen, right? C.elegans connectome until recently consisted of one animal, ever, right? And I can guarantee you, once we have the first connectome for a mouse, there's not going to be another $10 million or $10 billion spent on doing the EM connectome of 9 different animals. So EM connectomes have this annoying feature of having a very limited sample size, including C.elegans. So I'm not going to show you the EM connectome of a single UNC42 mutant animal that did show connectivity differences here because you're comparing an N of 1 with an N of 1. Not good. However, what I will show you, what we can quantify much better, is we can quantify the gene expression of putative synaptic adhesion and synaptic guidance molecules. We know a lot about them in these cells here, a whole bunch of ID domain proteins. We can quantify their expression level, and what we see, normally nice combinations expressed in individual neurons of that circuit. If you take away this Homeobox gene, the expression of those guys is very much disrupted. Here is looking at the very same problem with an even more telling class of molecules. These are molecules that form electrical synapses, the innexins. They're directly the constituents of electrical synapses. There's a whole bunch of them. We looked at 3 of them. Highly patterned expression within that circuit, responsible for these electrical synapses here. And again, their expression is very much disrupted in UNC42 animals. And again, that makes us to suggest that UNC42 is indeed required, not just cell autonomously to specify identity of these cells, but also to assemble them into functional circuitry. So now things get complicated, and I hope some of you may enjoy that, is of course the following question. Now, at first I was a little complex with combinatorial codes, and now I'm suddenly going to single genes. I'm telling you that this single gene specifies circuitry here, but these are different cells, right? UNC42 is required for all of them. If this were a class, I would ask you, well, how do you get different cell types within the circuit? Well, it's of course because there are different combinations of transcription factors that UNC42 works with, right? Let me take you through some of them. Here's a guy that works together with UNC42 to specify the identity of those 4, meaning makes those 4 cells become different from these other cells. Here's another guy that makes that one cell become different from those. These are all cofactors. These are all different combinations of transcription factors, and so forth and so on. So, but here's the interesting thing. The interesting thing is that each one of those transcription factors is not just expressed in that cell, but it again is expressed in a set of synaptically connected neurons. Before I show you that to you visually, which is going to look very confusing, I give you the punchline first. The punchline is, this is now boiling down this UNC42 circuit, these different cells, different neurons, synaptically connected. They all require that one transcription factor, UNC42, to be assembled into circuitry, we think, right? In each one of these cells, this guy is going to work with another transcription factor, and this other transcription factor is also expressed in a set of synaptically connected neurons, and so forth, and so on.
So that means that the connections are homotypic.
Sorry?
From this, it infers that the connections are homotypic?
Yes, that would be sort of one idea. One way to think about this would be that what those transcription factors do is they basically regulate the expression of, for example, a homophilic cell adhesion molecule, right? That brings those in proper neighborhood, connect to one another, right? And here's another guy that brings now this set of neurons together, and so forth and so on.
But that's not canonical, at least for the mammalian, like the thought leaders in the mammalian neural connectomic sphere, right?
They just don't have the resolution to make any of these conclusions, right? They don't have the cellular resolution based on anatomy, and they don't have the resolution based on gene expression. Let me just give you one more slide. Yes?
5 minutes.
Yes, and this is my last slide. No, second to last. So this is basically now showing you the real stuff here. So this is again in red, all of the UNT42 positive neurons here. In blue are these, this one overlapping transcription factor, a different circuitry here. And so you can envision that complex circuitry is built by the overlapping function of transcription factors that sort of link cells together into these overlapping circuitries. Now this is really my last slide, I promise. And now I want to sort of synthesize both aspects here, the Homeobox gene, preponderance, and the circuit association, right, into one sort of unifying idea, which follows this beautiful dictum here that has really characterized my thinking over the last decade or so. Nothing about biology makes sense except in the light of evolution. So maybe what we are seeing here is a reflection of the evolutionary trajectory of nervous system evolution, right? Meaning that if you think about the very first primitive nervous system, which of course doesn't exist anymore, think about a ball of cells, which were initially all homogeneous, and you now get the first two different cell types. These are generally thought to be a neuron-like cell that is becoming committed or dedicated to receiving signal and relaying signal to the other cell type, myoepithelial cells that generate some sort of contraction and therefore some movement of that ball of cell. Maybe this first set of neurons here, either by chance or by some specific biochemical properties of homodomain transcription factors, used homodomain proteins to specify these simple signaling features and their homophilic ability to be still attached to one another and talk to one another. But then also, of course, to secrete molecules to the neighboring cells to tell them what to do, right? So maybe this was the very first step here, and from there on, you get gene duplication and diversification, more homodomain proteins, their specificity may have slightly changed in terms of what genes they regulate. Different cell adhesion molecules came into being, regulated by different homodomain transcription factors, so you could maybe now see an individuation of circuitry. And then, of course, things became more and more complex, and of course, non-homodomain transcription factors came to play an important role as well. But maybe what we see here with the homodomains and defining these circuits are some sort of ur-circuits, some proto-circuits that are a reflection, a remnant, of the evolutionary history of these neuron classes. All right, so let me finish here and just thank again. I mentioned the people during the talk already. I just want to point out this was, of course, a joint effort by many students and postdocs over the years who did the self-made studies of Homeobox genes. But I want to again specifically highlight here at Yemeni for building this neuropal multicolor tool, which is really changing the way we can do things in the lab. And I mentioned those people along the way. Thanks for your attention. Yes?
Can you extend your claim even further and say something about functional connectivity, not only about physical connectivity, also functional connectivity, neurons that participate in a specific role in the worm and not necessarily connected? Let's say they are all responsive for a neuropeptide or...
Yeah, no, it's a great question. And we're just beginning with that. It's actually through the neuropal tool, and only through the neuropal tool, that we've been able to use GCAM to do real nervous system-wide activity recordings. And this, the recordings you can always do, but to make sense of nervous system-wide activity recordings, you could really say which neurons are active in response to certain stimuli. The problem is you'll have to do that with lots of stimuli to sort of see what are the different configurations, different states of the functional connectome, and then to correlate it here with Homeobox. We would love to do that, and we will eventually do that. Absolutely. Absolutely. Yes?
So in this transcription factor view of the connectivity, do you see
difference in motifs in connectivity? Like if you look at a particular transcription factor and the cells connected there and another transcription factor and the cells connected there, is there a difference in like some kind of connectivity motifs between them?
You mean the sort of the Oreo style little type of network motifs?
Maybe, or even more like large level statistical factors about
that. Yeah. You know, I'm sure we could dig deeper and analyze it. I'm again, you know, again, I say that again, I'm not shy to hide this here. We're basing that on a connectome with an N of 1. Right? So yeah, you may find some signal doing this sort of stuff. We've been a little reluctant to do that. It's very recent, only since last year, that more connectomes have come online. We're now getting sort of means to really boil down the core connectome of the worm, meaning where there's absolutely no variability seen across sex developmental stages. And this is actually not a trivial problem, but we're now getting to basically have a different connectome of the worm, and that will do hopefully this analysis then with this sort of more reliable connectome. Yes?
Sorry, I wonder if the expression of the different transcription factor is required or also required and sufficient to define. So one experiment that I don't know if you conducted, but would it be possible to knock down specific transcription factor and express others and reassemble neurons to new circuitry?
Yeah, so we've done exactly the second to last point, meaning taking away the terminal selectors that normally characterize the cell, put some other combination in, and this is very effectively reprogramming the identity of cells based on molecular markers. As to whether this has really also rewired neurons, this is not trivial to figure out. Essentially, you have to do, you know, a reconstruction of the connectome, right? I mean, it's you know, I mean, as you know, I mean.
No, I didn't say it's easy to do. It's easy to ask.
Yeah, yeah. No, we've very much thought about this, and this is of course sort of a prediction that we really were to reconstitute the codes and eliminating the initial one, and we can do that. We're just not there yet, technically, to do that.
Last question.
This was really fascinating. Thank you. Sorry. So I was really intrigued by this principle, regulatory principle that you also showed us, where there's this combinatorial regulation of multiple transcription factors, but there are these master regulators. I was wondering if this is actually the same principle when you look outside of the central nervous system. Is this like a general principle of, say, cell-fate identification, or is it unique to the central nervous system?
Yeah, no, it's a great question. You know, there are, I mean, there's a classic example, right? This is myOD, right? So in myOD, I mean, we now know it's not just myOD, it's a combination of 3 transcription factors, but this idea of having sort of a core set of transcription factors that specifies a neuronal setup is not restricted to the nervous system. It's not probably even restricted to any tissue, right? I mean, if you think about this, I mean, whether it's always going to be Homeobox genes is a different question, right? But the idea of having this coordinated control, right, is probably something we'll find in all tissue types. Which is really, you know, I really want you to appreciate this sort of, you know, every cell type, every mature cell type expresses dozens of transcription factors. So it's very easy to envision in principle that everything is chocolate, but it really says, like in the real world, not everyone is created equal. There's some people who are just more equal than others, and there are clearly some transcription factors that are really more important than others in a cell to specify the